Ready to go.
Hi, everyone.
All right, everybody. So this is a Bio 285 review, and it's with me, Jahan, and Syed. So we're gonna do this. So the first thing we're gonna talk about is DNA vectors. So like, what is a DNA vector? So it's basically a small circular piece of E. coli called a plasmid, and it's a vehicle to move around foreign parts of DNA in and between cells. So DNA plasmid vectors can be up to 3,000 base pairs long, and they can hold genes up to 20,000 base pairs long. And the vector must have an origin of replication. So for this is necessary for the organism because it's going to reproduce another cell. When it reproduces another cell, it has to have that origin of replication. So an example of this is E. coli plasmid have an E. coli origin of replication. So vectors must also have a selection marker, which we're going to talk about in the next slide. So vector selection markers. So when vector DNA moves between cells, not every cell receives the vector, so there must be a way to differentiate those cells who got the vector and those who didn't. So this is where the selection markers come into play. So they give the organism that received the vector a certain phenotype. So an example of this would be like the E. coli having antibiotic resistance, and they were using that as markers. And here, you can kind of see we have a plasmid map. So here you can see the antibiotic resistant gene, so that would be the marker. And then there'd be another selectable marker and the origin of replication, which is always necessary.
Excellent.
Yes. So next we're going to talk about restriction enzymes. So vectors need a way to be able to splice like the clone gene into the vector. So in order to do this, there has to be a restriction, digest, and ligation, and it's done using restriction enzymes and DNA ligase. So restriction enzymes or restriction endonucleases are DNA like scissors, and they're very sequence specific. So the restriction enzymes are made naturally in bacteria, and they are used in different experiments from bacteria because they're cloned and expressed and purified. So,
Oh, sorry.
no, you're fine. You can go to the next one.
Oh, okay.
So, splicing cloned genes to the vector. So, I put it out in steps. So, step one, your plasmid is going to cut with a particular enzyme. So, here in the picture, you can kind of see the plasmid and the cleavage site. And then the step two is they're going to cut a piece of the DNA that is going into the plasmid with the same restriction enzyme. So this would be here with the endonuclease, which would be our restriction enzyme. And step three, they're going to mix the cut gene and the cut plasmid with DNA ligase. And then in step four, when the cut ends, the plasmid and the gene will find one another and the DNA ligase will seal them together. And they seal them together by making a phosphodiester bond in the backbone. And what you definitely need to know is the minimal requirements for vector cloning are that it has an origin of replication, some restriction enzyme sites, and a selection marker. Those are the three things that they need. And then this is just another picture of cloning plasmids that I saw from the class that I thought could be helpful. And then I'm gonna hand it over to Syed.
All right. So now I'm just gonna go over gene isolation, and pretty much from this section, you're just gonna want to know the different ways of how you would go about, collecting a gene of interest or a plasmid, and then how you would make clones of that or make copies of that. Uh, and also down here, I have some like other videos that I found on YouTube. The first two are for PCR, and then the second one's for cDNA. The ones here are just like cool animations. I feel like it's helpful to actually see um, how these things play out rather than just like read about them or look at diagrams. Um, and then the one for cDNA is actually of this girl in the lab explaining how she goes about it. So for the first thing for sourcing of gene of interest, there's pretty much three ways you can go about it. One is you could find plasmid with the gene, or you could find the genome of the organism, right? If it's not too big, you can just go in and get your gene that way. Or the third way, say you have like an organism, for example humans, right? We have bigger genomes, right? You can actually go into genomic DNA libraries, which are organized into different plasmids, and you can pick out the one you want from there. Um, Yeah, so that's as far as you go for sourcing genes of interest. And then to make copies, you're going to want to do a polymerase chain reaction, which is literally in the name. It's just a reaction that goes on and on, like it repeats itself. Um, Basically, the goal is here, you're going to want to take the gene, right, the DNA sequence that you want to test. Then you're going to want to unravel the two strands and then throw in some primers to replicate each end, and then just repeat that process just to make more and more copies exponentially, right? So you're going to go from like 2 to 4 to 8 and so on. And so for the prep for this is you're going to want to order your plasmids, right? And they're going to need to be complementary to the DNA strands that you're going to be targeting. Um, and these like you can order like from any company, but you're just going to want to know that of course, the plasmids should be from the 5' to 3' direction, and they're going to want to run anti-parallel, right? So they're going to run the opposite direction for DNA transcription. And then the second step, you're just going to want to take the DNA or the gene that you want to look at, the plasmids, the DNTPs, which are just DNA bases, right, to actually add on to make the new strands, and then, of course, DNA polymerase. Um, and then you just put that into your reaction, and then you start thermal cycling. And thermal cycling is the process of how you're going to want to copy these um, DNA sequences, right? So the goal is that in about 20 to 30 cycles, you're going to have millions of copies from your original DNA strand. So basically here, you just want to know 3 steps. You're going to start off by denaturing DNA at 95 centigrade, right? That's going to break the hydrogen bonds between the two strands and unravel the DNA into the 2 separate strands. And then the second step, you're going to want to annul the primers right at 50 to 65 degrees centigrade. So you're going to cool down the temperature. You're going to give the primers a chance to go in and bind to the 2 different strands, right? And one thing that you may note of is that the temperature for annulling the primer is going to be dependent on the primer itself, right? So you don't want it to be too hot, whereas nothing's going to bind, and you don't want it to be too cold, because at that point, then the primers aren't going to be as specific to what they prime or to what they parity. So you're going to want to, depending on what your primer is for whatever sequence you have, you're going to want to optimize that temperature. But usually it will fall between that. And then the third final step is you raise the temperature again to 75 to 80 degrees centigrade. And the reason for that is that you're going to be using Taq polymerase, right? So you're not going to be using the normal DNA polymerase in eukaryotic cells, because at that high of a temperature, it will just not function. But luckily, if you use a Taq polymerase, and it goes into more detail of that in the 2 videos earlier, um, basically the Taq is going to be optimized to be able to function at that temperature. And then you have the diagram here from a video that just shows you how repeating the cycle is going to just grow the copy. And on to restriction sites, right? Say you have a vector and you want to add a restriction site to it so you can cut at a specific point. So basically, you're just gonna uh, go to the 5' end of your DNA sequence, and you're gonna add a restriction site to that end, plus some extra DNA bases, and that's just to help when it cuts, it's gonna be clean. Uh, right, and then you're gonna want to take that PCR product, right, the one now that you have with your restriction sites, and you're gonna cut that together with the plasmid that you have of interest. Um, and basically, like you went over the video, right, you're gonna have here your product and here your recipient plasmid, and you're gonna have, say, for example, here we have these two restriction enzymes. You're gonna digest both, which is gonna give you complementary pairs on each end, right? You got your AATC, and then you got the other end here, and so just the same for the other. Now you have your GC and you have your CG, and you're gonna want to put these together in with a ligase so they connect or they come together, and then there you have your finished product with your plasmid that contains the restriction sites, and that's pretty much that. Also, if you guys have any questions, feel free to just unmute yourselves. But if not, I'll keep going. And then the last thing here is we're going to have our complementary DNA or cDNA. And so basically the goal for this is that, say you have a eukaryotic gene, right? A lot of ours are going to come with introns that, luckily for us, we can just splice out. We have the machinery for that in our cells. But if you're going to be putting these into an E. coli, right, into a bacterium, it's not going to have that because they don't even have introns they have to deal with. So you're going to need to find a way to figure out how to like splice out those introns and then put it into the E. coli, right? So that's where cDNA comes in. It's basically a way to take mRNA product from your gene of interest and then use that to reverse transcribe the full gene, uh, of course, without the introns. So basically the procedure for that is you can use affinity chromatography, and that basically just pairs up nucleotide bases of your target mRNA, right, via its poly A tail. So it's going to go to that tail and it's going to pick that out. And then you can just wash away everything else. So you're just left with your mature mRNAs. And then you're going to incubate them with a reverse transcriptase, right? So that's going to make the complementary base of your mRNA or the DNA version of that. So now you have your single strand DNA. So then you're going to want to incubate that with a DNA polymerase and a terminal transferase, and that's basically just going to create the other side of that double strand. And then it's also going to add the single strand tails. So at the end of that, oh, is that the, oh, I thought I had a figure, but I guess not. It was just the same figure as her one from the video. But basically, right, you're going to have, after these 3 steps, you're just going to have single or double strand DNA that you can then put into your vector. And the important thing to note there is that you're going to have your DNA strand without the introns because they were already spliced out when the cell made its mRNA. But yeah, that's pretty much all we have for that. If you guys have any questions, feel free to speak up.
We're gonna post the recording for this onto the class YouTube as well.
Yeah. So if you missed anything or you want to go over anything else, you can just watch that.
But thank you guys for coming.
Yeah, thank you guys so much. We can stop.
You're welcome, Sophia and Austin. You're welcome, Emma.